It's been hours since you last ate. Your blood glucose is starting to dip, but your brain needs a steady supply of glucose every second to keep working. So where does that glucose comes from between meals? From the store you built earlier. Your liver has been holding a reserve of glycogen and now it starts breaking it back down, releasing glucose into blood to keep you going. This breakdown process is called glycogenolysis, and it's the exact reverse of glycogenesis we built last time. But it's not simply glycogenesis run backwards. The cell uses completely different enzymes to take glycogen apart, and it uses a clever chemical trick to break the bonds without spending water or wasting energy. So in this video we will go through every step, and at each one we'll look at the actual structures, how the chain is cleaved, how the branch points are dismantled, and what finally comes out. So let's begin. So glycogenolysis is the catabolic reaction for the breakdown of glycogen in the cell. It is the breakdown of glycogen into glucose or glucose derivative molecules. Note that carefully. Glucose to glucose derivative molecules. As we will see, most of what comes out isn't free glucose at first, but a phosphorylative derivative. Hold that thought. Why does the body do this? Because the process is essential for maintaining blood glucose level, especially during period of fasting or increased energy demand. When you are exercising hard, glycogenolysis releases the stored glucose to keep your blood sugar stable and your muscle fueled. A quick recap of what we are breaking down. Glycogen is polysaccharide composed of glucose units, and it is stored primarily in the liver and muscle. Remember its structure from the last video. A huge branch molecule. Long chain of glucose linked by alpha 1,4 bond. It is the straight run. And the branch points linked by alpha 1,6 bonds, roughly every 8 to 12 residues. Many non-reducing ends at the tip of the branches, and these ends are where breakdown happens. So the strategy is two enzymes and they have two jobs. Glycogen phosphorylase. It chews glucose unit off the straight alpha 1,4 chain, but it gets stuck near branches. The debranching enzyme dismantle the alpha 1,6 branch point so phosphorylase can carry on. Now let's talk about the structures. Step 1. Phosphorylasis by glycogen phosphorylase. This is the main event, the reaction that releases the bulk of glucose. The structure going in is the glycogen chain. Look at the non-reducing end. The terminal glucose whose carbon 4 hydroxyl is free. The terminal glucose is joined to its neighbor by an alpha 1,4 glycosidic bond. That is our target. The enzyme glycogen phosphorylase cleaves that terminal alpha 1,4 bond. But notice how it does it. It does not use any water. It uses inorganic phosphate. This is called phosphorylasis, bond breaking by phosphate, and opposed to hydrolysis, which is bond breaking by water. Why does that matter so much? Because of what it produces. The structure we produce is glucose 1-phosphate. As the phosphate attacks the bond, the released glucose comes off already carrying a phosphate on its carbon number 1. It's glucose 1-phosphate, not free glucose, and the glycogen molecule is now shortened by one glucose residue. Think about it. If glycogen were broken down by hydrolysis, you'd get free glucose, and to do anything useful with it, the cell would then have to spend an ATP to phosphorylate it. But phosphorylasis delivers the glucose already phosphorylated for free using cheap inorganic phosphate instead of ATP. So the cell recovers its stored glucose in an activated form without spending any ATP. That's the whole point of using phosphorylasis. Glycogen phosphorylase keeps working along the chain, snipping off one glucose 1-phosphate after another from the non-reducing end, producing many glucose 1-phosphate molecule in a row. But it hits a wall. Glycogen phosphorylase can only cut alpha 1,4 bonds, and it cannot get close to a branch point. It stops 4 glucose residue away from an alpha 1,6 branch, leaving a stubby, still branch structure called limit dextrin. At this point, phosphorylase is struck, and the second enzyme has to step in. Step 2. The debranching enzyme. The debranching enzyme is remarkable. It is a single enzyme with two completely different catalytic activities, and it uses both one after the other to clear away a branch point. So let's take them in order. After phosphorylase has stalled, we have left with a short branch, typically 4 glucose residues attached to the alpha 1,6 branch point. The transferase activity of the debranching enzymes picks up a block of 3 glucose unit from the branch and transfer them onto the nearby non-reducing end of the main chain, reattaching them there through a new alpha 1,4 bond. The structure we produce is those 3 glucose are now added onto a main chain, extending it, and the branch point has been reduced to a single glucose unit, still attached the branch point by alpha 1,6 bond. So the transferase has tidied up the branch, moving most of it back onto the straight chain where phosphorylase can eventually reach it. Now there is just one glucose left, clinging to the branch point by the stubborn alpha 1,6 bond. The bond phosphorylase could never touch. The alpha 1,6 glycosidase activity of the same debranching enzyme hydrolyzes that remaining alpha 1,6 bond, cleaving off the last branch point glucose. And here's the key distinction from step 1. This reaction is a hydrolysis. It uses water. So it releases a free glucose, not glucose 1-phosphate. So overall, glycogenolysis produces two products, about 90% of glucose 1-phosphate and about 10% of free glucose. The structure we are left with is, with the branch removed, now we have an unbranched alpha 1,4 polymer, a clean straight chain. So the two enzymes work as a team in a repeated cycle. Phosphorylase chews down a chain unit it stalls at branch point. Debranching enzymes clears the branch, and phosphorylase resumes it, and so on, dismantling the entire glycogen molecule piece by piece. So what happens next? What is the fate of the product? So we have released mostly glucose 1-phosphate, but glucose 1-phosphate isn't the cell's usable currency. So what happens to it? This is where the story spills and where the liver versus muscle difference becomes crucial. The glucose 1-phosphate is converted to glucose 6-phosphate by enzyme phosphoglucomutase. The phosphate is shifted from carbon 1 back to the carbon number 6. This is the exact reverse of a step you saw in glycogenesis. The same enzyme runs both ways. Now the path diverges. In the liver contains a special enzyme, glucose 6-phosphatase, which removes the phosphate from glucose 6-phosphate, producing a free glucose. This free glucose is then released into a bloodstream to raise the blood sugar level of the whole body. This is why the liver is body's blood sugar regulator. Only it can convert its glycogen back into free blood glucose for another organ like brain to use. In the muscle is different. Muscle lacks glucose 6-phosphatase. It cannot remove that phosphate and therefore cannot release free glucose into the blood. Instead, glucose 6-phosphate is fed straight into glycolysis to generate ATP for muscle's own contraction. So the same pathway, glycogenolysis, serves two different purposes depending on the tissue. In the liver, it feeds the whole body blood glucose, but in the muscle, it fuels only the muscle itself. This liver versus muscle distinction is one of the most important and most tested. Let's talk about the regulation and significance. So hormonal control. The key regulatory enzyme is glycogen phosphorylase. It is controlled by reversible phosphorylation by glucagon and adrenaline epinephrine. Both switches glycogen phosphorylase on, driving backwards. And insulin does the opposite. It switches the breakdown off and favors storage. This is reciprocal regulation. The same hormonal signal that turns breakdown on simultaneously turns the synthesis off. So the cell never wastefully does both at once. Now let's just quickly recap. Step 1. Phosphorylasis. Glycogen phosphorylase cleaves alpha 1,4 bond from the non-reducing end using inorganic phosphate, releasing glucose 1-phosphate, saving 1 ATP. It stalls at 4 residue from a branch point, which is limit dextrin. Second step, debranching. The debranching enzyme uses two activities. Transferase. it moves a block of 3 glucose to main chain, leaving 1 glucose at the branch. Alpha-1,6 glycosidase hydrolysis that last glucose off, releasing free glucose. The result is an unbranched alpha-1,4 chain, ready for more phosphorylase. Third, fate of the product. Glucose 1-phosphate converts to glucose 6-phosphate by phosphoglucomutase. Then liver has glucose 6-phosphatase, which frees the glucose into the bloodstream. In muscle, glucose 6-phosphatase is not present. So glucose 6-phosphatase directly goes to glycolysis and forms ATP for itself. So the final products we've got is 90% of glucose 1-phosphatase and approximately 10% of free glucose. And these are controlled by both glucagon and adrenaline. Two enzymes working as a team dismantles the whole store, and phosphorylasis makes sure it's done without wasting any energy. So if this logic of glycogenolysis clicks now, why the cell uses phosphatase instead of water, why muscle can't share its glucose but the liver can, that's what our channel is for. Practice drawing the pathway with structures and revise it side by side with glycogenesis. They are the mirror images. So that's it for this video. Keep learning, keep achieving. I'll see you in the next one. Thank you.